Buenas a todos, mi nombre es Carlos Pagan, soy un profesor asistente del Departamento de Patología y Cell Biology. La presentación es en inglés, pero cualquier pregunta en español, muy bienvenida. I have nothing to disclose regarding the presentation. I have no financial ties. So I wanted to start the presentation on molecular pathology and molecular testing by giving you a brief idea of what molecular testing has gone through in the last few years. So, even the earliest we have of the structure of DNA itself as a duplex, double helix is from 1953. In the 60s, we started introducing the first techniques to even look at DNA in a testing setting with hybridization techniques and electrochemical detection of DNA. In 1975, we started doing southern blotting, which is a big advancement for us in genetics. And in 1985, we have PCR, which is the most common form of genetic testing that we had until most recently. And in 1995, we finally sequenced one bacteria, which was a big breakthrough at the time, but now it seems kind of small. And that was about 101.8 million base pairs. And compare that, I'm going to leave the story for now, on the year 2000, when the first versions of the Human Genome Project were released, that basically had about 3 million base pairs and about 20,000 protein coding genes. So in the context of that, how does this will apply it to cancer? But I wanted to go a little bit into what is cancer as we think about molecular. And in the term of pathology, cancer is just an uncontrolled proliferation of cells. It's rapidly proliferating, not losing all its self-regulatory control. But cancer is also not one disease, it's actually a family of diseases. Each cancer has its own context. Lung, colon, breast, these are all very different diseases on themselves, even though they share the name cancer. And cancer is also a genetic disease. It's a disease that's caused by mutations in somatic cells. So that's the actual underlying cause of all cancer. Now, to go a little bit into what it means to lose control in a cell. In a normal cell division, you'll have normal cell proliferation. Your cells will proliferate as normal, but they may occasionally encounter some cell damage. Your body has ways of detecting this damage and correcting it by destroying those cells either through suicide or apoptosis. But in cancer, those mechanisms go out the window and they basically accumulate mutations, a couple at first and then many more, until these populations completely lose the ability to regulate themselves and create a tumor. And it's a pretty big problem, especially in the US. About 800,000 men and women will be diagnosed with cancer in a given year, and these can be very highly prevalent. Some cancers are very frequently seen, like breast or prostate or lung and colon. And there are molecular techniques that we can do to kind of help along these different tumors, which we'll discuss in a little bit. Before that, I want to give you an idea of what we test in molecular cancer testing, and that's oncogenes. Oncogenes are just very limited, in a limited way, a gene in which, in certain circumstances, can transform a cell into a tumor cell. Now, oncogenes come in two essential types. Basically, oncogenes, as we think of them as activating genes, proteins that become activated and induce proliferation, versus tumor suppressor genes, genes that will stop cancer when they're acting normally. And then we test, I mentioned earlier, these are mutations in somatic cells, and somatic cells just means cells that are all over your body but are not inherited into children. So that's in contrast to germline mutations, which are mutations that are inherited. An example of an oncogene, as we think of them, is, for example, KRAS that I'm mentioning here. So KRAS is a very frequently mutated oncogene. It serves, I'm using it as a prototype for an activating gene, where you can see here that this model of the actual gene itself. There's many mutations we can detect, but the ones that cause cancer are kind of located in this region of the tumor. So this. In contrast, you have tumors like you have tumor suppressor genes like TP53, which is the most common gene mutated in cancer, and you can see that it's mutated throughout the entire genome. So it would require us a lot of sequencing to identify all potential mutations in one of these genes. And this has only showed you two genes, but there's a huge amount of genes that can be involved in cancer, and they can have huge range of effects, including growth factor genes, genes that control cell cycle in the cells and mitosis, genes that control telomerases to keep cell replication and mortality, apoptotic regulators like TP53, and many others that we haven't even discovered yet. But this relates to how cancer progresses in mutations. So in the progression of cancer, you begin with your normal tumor cells, and as you accumulate a mutation, you'll advance to a precancerous lesion, and you'll acquire a few mutations there. You'll acquire a few more, and you'll end up with the stage 1 cancer, which is a cancer that's localized, small tumors. As you get more mutations, it might become more locally advanced, spread to lymph nodes. You'll have more late locally advanced, with lymph node involvement, and then eventually you ultimately reach a stage where the cancer has accumulated enough mutations and enough aberrant functions that it metastasizes and can spread throughout the entire body. So what does pathology serve in the treatment of cancer? So pathology really works as an information center. Our job is to basically take tissue and spit back a result. So clinicians in the community will just give us something, and we'll try to give them something they can use. We want to be something usable and provide meaningful information for clinicians. The diagnosis of cancer really relies on tissue. So we need to have tissue that might be a biopsy, a surgery. Those are the kind of things that we need to sort of establish establish diagnosis of cancer. And what does pathology do in this sort of precision medicine that we're trying to achieve? Well, pathology basically is the lab. It's that report, those results that can do it. So in the typical setting, a patient will go to their doctor and perhaps be diagnosed. They will be referred to the surgeon or to whoever will take biopsy, and that'll be sent to pathology. And in the lab, we'll basically submit that tissue to a lot of formalin. We'll put it into wax and put it up into little pieces, like you can see here, little tiny pieces, and then put it into slides, and we'll look at those slides in the microscope, and you'll get something kind of like this, where we can diagnose cancer based on the features of this under the microscope. So these are just an uncontrolled proliferation of cells. This is, clustered. They have no architecture. They have, large nuclei. They look very atypical. And not only that, now we can also do immunohistochemical stains for proteins, so we can find out what the proteins are doing in this tumor. So, for example, this is a stain for an ALK protein. So, because it stains highly for ALK proteins, it means that it's an overexpressed oncogene in this thing, and we can basically treat this patient for inhibitors against ALK. Now, molecular pathology, I pulled this straight from the Wikipedia article, and it's basically an emergent discipline within pathology, which is basically studying the diagnosis of disease, which is just pathology in general. But in molecular, it's through the examination of molecules within tissues and bodily fluids. Essentially, I can just summarize and say molecular pathology is pathology that tests for molecular biomarkers. And biomarkers is just these small molecules we can find in the body. And these can either be predictive or prognostic. Predictive meaning that they can tell us how the tumor will behave, or they can be prognostic, meaning that they can tell us what we can do, how they will respond to different treatments and different chemotherapies and different agents. And it can be kind of tricky to keep your mind around what does this mean, why is this useful at all? But there's a lot of guidelines that have been developed in the last few years about how to manage this information. So, for example, in lung cancer, there's guidelines of molecular testing, what tumors should be tested, and what genes should be tested, and why is it important, because we can give them a tyrosine kinase inhibitor and treat these patients and hopefully prolong their lives. And there's guidelines for all kinds of organs, like colon, pancreas, breast. There's a lot of them, and they can be very tricky, and they're constantly changing. So pathology can help here as a really useful resource, along with oncology, to sort of determine how best to manage these patients. And there are lots of drugs. Some of these have existed for a long time, like imatinib for CML. It's a very well-known, probably the first example of precision medicine for using imatinib, but we also have things that are for very frequent cancers, like breast cancer with trastuzumab, the ALK I mentioned earlier with crizotinib, and even melanoma and lung cancer can have potentially treatable targets with these drugs. And so these drugs exist. We in pathology can test for them, and that's how, that's our role in precision medicine, to give the appropriate treatment with the right drug and the right gene. And this can even extend into patients who have germline mutations, in patients who might have had a germline risk, an increased germline risk for cancer. Now can actually have drugs that target that risk and treat these patients in a way that meaningfully extends their lives. And the reason we can test these patients now and frequently in the lab is because the cost of genomic sequencing has really gone down dramatically in the last few years. I stopped the last history of molecular about the year 2000 when the human genome came out, and at that time it was about, to give you an example, it's about $100 million to sequence one human genome, and now it's basically we could sequence a human genome for less than $1,000, and we do it all the time in any of these buildings in Colombia. So it's really much more accessible to have sequencing done, and sequencing on a giant scale is possible. So because as molecular pathology has grown in the last few years, because we went from testing single genes, PCR from technology from the 80s, to now using very recent technology, which is massive parallel sequencing, or as you might hear, next generation sequencing, where we can test hundreds and thousands of genes in a very relatively short amount of time. And just to give you an example of what we actually experience in the lab when we see this, the picture on the left is basically what it used to be when we would look at genes. So each little line in here is a nucleotide. So each one of these is an A, T, G, or C, and you could have to count all of these with a ruler and go down each line. And that was not that long ago. Nowadays, this is a glass slide. It's about the size of my thumb, and if you look at it microscopically, in that slide you'll have millions upon millions of little probes in it, that each of them carries a DNA strand, and each of them will highlight in a cycle with the color of their nucleotide. So you'll take a picture of the slide at this magnification, for example, and the green can mean A, and blue can mean C, and you can basically do this work in no amount of time. Doing this sequence is about 50 nucleotides. It could take you about 3 days. We could do millions of nucleotides in 3 days now too. So it's a significant technological increase. And there are different ways we can do this next generation sequencing technology. We can either do PCR, similar to what we had before, where we have we can do targeted genes. Only test, let's say, 50 genes, for example, which is very the clinically relevant genes. We could do whole exome to try and discover new genes that might be involved in cancer. We could do whole genome and just survey the entire genome, and that can give us a lot of insights. And we're doing this to sort of learn more and kind of reach the limits of what molecular can do. And once you're tired of DNA, you can jump to RNA and start testing the transcriptome as well. You can test either the exons, or you can test the whole transcriptome for a patient. And now we can even do this on formalin-fixed tissue. So patients who had cancers and they were sent to pathology many years ago can now be tested even if without fresh tissue, which used to be also a limiting thing before. So our ultimate goal is that when a patient has cancer, whatever it may be, we send it to pathology. We'll look at it under the microscope. That's our gold standard. But we can also subject it to clinical molecular testing. And with molecular testing, the goal is that we can say, oh, well, this patient has this mutation. We can give them drug A, or this patient has this mutation, a different mutation. Let's enroll them in a different clinical trial that doesn't exist. Or this patient has this mutation, and we can say, oh, this cancer will behave in a certain way. And it's a lot of information. It can be impossible to keep up with it. So there's a lot of resources we can use, especially for clinicians in the community can use clinicaltrials.gov to find a person, if you have a patient with a particular disease condition, like say lung cancer, and they say, oh, well, I have a report that says they have this mutation, maybe I can find a clinical trial for them and hopefully meaningfully extend their lives. And so at Columbia, we do a lot of this testing. It's a kind of a shameless plug for our department, but I'm really proud of what we do at our work. So we can do testing that's very targeted, a couple of genes, the V600F for your polycythemias, or we can do all the way up to whole exome sequencing and do lots of gene panels. So we really want to provide a service that encapsulates the needs of any patients that come to us. And and it seems slow, but to us it's really a big deal that about 36% of the mutations that we find are actionable will have a meaningful change to our patients. So about 36% means that the patient will get a new drug, the patient will get a different chemotherapy. So for us it's a really big deal that we can get that amount of change. And and earlier I said, oh, you can't, you have biomarkers in the blood, and you can't diagnose, you need tissue diagnosis. But now really, the technology is reaching a point where we can actually do a liquid biopsy and do molecular testing just on peripheral blood. We have our liquid, the concept of a liquid biopsy, where a tumor will shed DNA into the bloodstream. They will release this DNA either through apoptosis or through necrosis, or just basically out of proliferation. DNA will be released into peripheral blood. And then we can extract that DNA, and if we know the mutation is characteristic for a tumor, we can say, well, this mutation, the person has this mutation in their blood, they likely have this tumor, and we could, in theory, test it and maybe find it early and screen for these things. We're not there yet, but we can find the mutations that are relevant. If they have a lesion that may be difficult to biopsy, it's much easier to draw a blood sample at your office and just send it for molecular. With liquid biopsy, you can do that very easily. We can also use it after you resect the tumor. So if a patient has cancer, they get resected, and they're doing okay, you can every once in a while do a blood test and see if that cancer is coming back and try to find it as early as possible. So these are the kind of the technologies that we're going to see in the future much more frequently. And as tumors become more aggressive and more resistant to therapies, we can still keep track of them and hopefully find better ways to treat them. So this kind of fits with the model for precision medicine, which is that a patient will be diagnosed with a cancer, but not every lung cancer will be the same, not every colon cancer will be the same. So you really have to take into consideration the patient's history, their race, their family history, their gender, their lifestyle, what they're exposed to, and then combine that with their genetic data and try to find the most appropriate and best therapy for that patient. So that's our goal as in precision medicine, and molecular pathology want to be part of that as well. So I want to also kind of give you an idea of how this works in practice. I'm going to give you a bit of a case that I had many years ago that's kind of really was impactful for me, and I wanted to share it. So this is a 36-year-old woman who was diagnosed with breast cancer at 36. She's a healthy woman, no medical history, no family history of cancer, but she did find a left mass in her left breast at about 12 to 3 o'clock, so like in the upper outer quadrant of her breast. And from the imaging, it's summarized, but it's a sizable mass at 36, so it's very worrisome. When we get the tumor in pathology, we see these cells here that are kind of clusters of cells arranged in cords, and they're very atypical, like the picture I showed you before. So this is an adenocarcinoma. It's invasive. It's in this fibrous tissue here. And on the right, you can see that this is a vascular space. This is like a vein or a lymphatic duct, and there's cancer inside of it, so this is what we call lymphovascular invasion. And she's 36. She has an aggressive tumor, so we want to treat this patient and try to give them the best therapy we can, because we want to preserve her life, and hopefully, she's 36. We want to be aggressive against it. So she underwent our typical pathology workup. So she has, this is the protein workup with immunohistochemistry. We did ER, so estrogen receptor, and PR, progesterone receptor, and those had some things. So she already qualifies for hormone therapy with trastuzumab. And then we also tested for HER2, which is HER2 is basically a protein that can be tested with different antibodies. Sorry, I said trastuzumab for ER. I meant tamoxifen. For HER2, you can treat it with trastuzumab. It's an immunoglobulin, but in this case, it was equivocal. It wasn't high enough to qualify, so this patient wouldn't respond to that therapy. So we're basically limited to treating her with trastuzumab. But then, she had a mastectomy, so she had that tumor out. And then with that lesion, she underwent molecular testing. In this case, with sequenome, it's a smaller panel, but we still found a mutation in ERB2, the V777L mutation, which is it's a mutation in the HER2 gene. And this is basically a representation of HER2, the gene itself. This is the extracellular domain of HER2, the juxtamembrane domain, the tyrosine kinase domain, and the tail that's not as much active. And you can see that the patient's mutation here has been documented before, and we can tell that this mutation is an activating mutation in HER2. So that's potentially, even though she doesn't have an amplified HER2, she can potentially be treated because she has an overactive HER2. While this is happening and we're doing the molecular testing, she gets additional screening, and we find that she has a lymph node mass here, and she has a mass in her pelvic bones. So when we biopsy these masses, we find the same tumor that's in her breast. So she already has metastasis. Again, she's 36, so the HER2 probably is explaining why this tumor is so aggressive, because it has a very active protein that shouldn't be doing that. It's dysregulating her. So now we know that she has a mastectomy original with the HER2 that's not helpful, but we know that it has a mutation. But when we test her metastasis, she has that same mutation in both her iliac bone and in her axillary node. So we know that it's truly a metastasis because they share mutations. But something different is that she has a HER2 amplification here. So this basically, she has a mutation that makes it hyperactive and an amplification. So she has more copies of these genes than she should have. So she has kind of a double hit for this gene that is producing her cancer. So then, as part of our, in pathology, this is a difficult case. We want to treat her well. She was then enrolled in this trial, the 286 trial. It's a phase 1 trial for a drug. This is not the typical thing people go through when they get cancer, but this is kind of the things that are possible when we do molecular testing on these patients. And we enrolled her into this trial with this drug that I'm not going to bother, but basically for patients with advanced cancer. And this drug is supposed to treat patients who have an overexpression of HER2, which we now know she does have. She is hormone receptor positive, which she is, and she has mutations in this line of PIK3CA, which is basically where HER2 is. And so, looking at her molecular and her cytogenetic context, we can see that this is probably would be helpful for her. And it turns out that it actually was helpful for her. When we gave her this drug after therapy, she was treated and her bone mets kind of went away. She had a sternal mass that was discovered afterwards, and it also kind of went away with therapy. And then her axillary node also went away. So this is an example of a really great success in therapy with this patient. I give you this very dramatic response. That's kind of what we hope to see in cancer treatment, but I have to remind everybody that this is not a miracle cure, and even this cancer that responded so well eventually came back later, many years later, when she developed resistance to these things. So it's a very difficult disease, but this can significantly prolong her life. She can spend more time with her children. She can be with her family. So that's what it's very important for us to keep that in mind when we treat cancer. It's not a single thing. It's not a single one thing will cure the patient, but we can provide meaningful changes for her. And then finally, I want to re-stress the importance of working as a team. For a cancer diagnosis, it takes it takes a lot of information. The clinical team has to see the clinical history. We can provide genomic analysis with sequencing and all these different techniques we use in the laboratory to find meaningful data. Once we have that, it can be very difficult, finding her the right trial, what are the right drugs to use. Oncology and pathology and clinical teams work a lot for this, and hopefully we'll have a good outcome when the patient responds, and we'll do well. And finally, in summary, I think I want to say that molecular pathology is just an addition to the work we do in pathology to try and assess tumors and give diagnoses, and that molecular can provide really useful information prognostically and therapeutically, and we have a lot of guidelines that can help us with that, and that making treatment decisions in these patients with cancer is really a multidisciplinary approach when we have to include all members of the team to come to the right diagnosis for these patients. And that's